Namaste teachers. Welcome to biology session. Are you looking to refresh your teaching plans? Do your students find the heredity chapter particularly difficult? Are they often confused when writing phenotypic and genotypic ratios for monohybrid classes? Are they struggling to draw Punnett squares? If so, this video may be helpful for you. Objectives. By the end of the session, teacher will be able to refresh their teaching plans, recall and connect the steps in the period plan to their own plans, gain ideas for addressing challenging concepts in the chapter on heredity for class 10. In this session, we are going to see a model lesson on difficult concepts from the chapter heredity for grade 10. Before going into the model lesson, let us recall the steps to be followed for a classroom transaction. For the fruitful classroom transaction, we should work in 3 stages: pre-teaching, during teaching, and post-teaching. In pre-teaching stage, we need to perform 3 major activities: study, plan, and collection of resources. Study. We may have extensive knowledge of the concepts. We may have years of experience with a particular class, but we may still overlook important aspects of the subject matter sometimes. If we take the time to study these concepts beforehand and gather updated information on the topics, it will boost our confidence during class and make our sessions more engaging and productive. Therefore, let us commit to continually learning and updating our knowledge about the concepts we teach. As Rabindranath Tagore said, a teacher can never truly teach unless he is still learning himself. A lamp can never light another lamp unless it continues to burn its own flame. After study, we have to plan. A good plan is like a roadmap. It shows the final destination and usually the best way to go there. After studying, we need to plan the classroom transaction for that specific concept. We should determine how much time is needed, what resources are required, where we can obtain them, and which strategies we should adapt. All of this should be organized before the classroom session. To facilitate this, we need to prepare a yearly plan, a lesson plan, and periodic plans. Failing to plan is planning to fail. SCRT's academic calendar provides a general framework for planning classes. Now, SCRT plans to provide teacher handbooks containing detailed information needed to plan effectively. So let us utilize them. Then, collection of resources. Gathering resources before entering the classroom is crucial for our success. Even with the thorough planning, it's essential that all resources needed for visualizing concepts are prepared in advance. Teaching a class without teaching learning material is just like fighting a war without weapons. When we utilize resources effectively, our classes become more engaging, encourage greater student participation in activities, and enhance the overall impact. Then, what we have to do during teaching? To improve teaching effectiveness, we should focus on engagement, clarity, and adaptability. Using interactive methods like discussions, group work, and real-life examples, we make lessons relatable and promote critical thinking. Effective techniques such as multimedia tools and simplifying complex concepts can enhance understanding. Encouraging students' participation in a welcoming environment and utilizing various questioning techniques along with peer-to-peer learning will further boost engagement. Recognizing different learning styles is essential. We should offer diverse materials and differentiated instruction to meet varying abilities. Continuous assessment and constructive feedback will guide students' improvement. By adopting these strategies, we can create an engaging and effective learning environment that maximizes the student's success. Then, what about post-teaching? After teaching, we should assess the students to understand their progress. Based on the assessment results, we can provide guidance, monitor their development, and adjust our remedial teaching strategies to support their improvement. The teacher's role is essential in these three stages of learning for ensuring the success of the student. When it comes to teaching period transaction, there is no single template that fit all classes, subjects, and concepts. These templates can vary depending on the subject, class, and the specific concept being taught, as well as the nature of the students and the objectives of the lesson. However, to maximize the student learning, we can classify lesson plan templates into three categories. 1. The first teaching period of the chapter or unit. 2. Subsequent teaching periods for the chapter. 3. Remedial or practice period. First teaching period of the chapter. This typically consisting of several steps. There are 12 steps in this period template. The first one is greeting and introduction. The teacher starts the lesson by greeting students and creating a friendly learning environment. This step helps engage the students and prepare them for learning. The second step, announcement of the name of the lesson and a topic. The teacher introduces the lesson title and topic. This provides clarity on what students will be learning. Then mind mapping. The teacher creates a mind map to visualize key concepts. This helps the students connect new ideas with prior knowledge. Then the fourth step, prerequisites. It's very important, crucial. The teacher assesses the students' prior knowledge and motivates them to connecting the topic to real life examples. This ensures a smoother learning experience. Then the sixth one, model reading. You may wonder why a science teacher has to do model reading, why a science teacher has to make the students to read the text and find out the keywords. But remember, as we are gradually transforming our education system from Telugu medium to English medium, it's the responsibility of each and every teacher to support the students in learning English and gradually develop their skills in English so that those language skills helps to learn the content in the other subjects also. So using the biological text, we can make our children learn the English vocabulary and uses of the English language. So here, these steps were also included. Model reading. During this step, the teacher reads the lesson aloud, demonstrating correct pronunciation and expression. This step helps the students understand how to read fluently. Then reading the lesson and identifying keywords by the students. Students read the lesson and pick out important words. This improves their reading comprehension and vocabulary. Writing keywords on blackboard and discussing their meaning. Keywords are written on the blackboard and their meanings are discussed. This reinforces the understanding and helps in retention. Then we will come into the actual very crucial important step of the period plan. That is understanding concepts through activities or pedagogical strategies. The teacher uses activities, experiments, and other methods to explain the concepts. This makes the learning interactive and engaging. Then presentation of activities and discussion. Students present their understanding through discussions, role plays, and presentations. This step enhances the communication skills and deepens the comprehension. Then, summary. Different strategies can be adopted to summarize what the concepts are discussed in that particular period. One of the suggested better way of summarizing is students will summarize what they have learned and the teacher reinforces the key points. This ensures that students grasp the main ideas. And finally, assessment. The teacher evaluates the student's understanding through quizzes, questions, or other assessment tools. This helps in identifying learning gaps and providing necessary support. Each step in this period plan plays a crucial role in making the lesson structured, interactive, and effective for students. These steps align with the 5E model also: engage, explore, explain, elaborate, evaluate. Actually, the 5E model is an inquiry based instructional framework that aligns well with the steps in the provided template here. If you see these 5 steps, they engage the student in the teaching learning process. And these 3 steps give scope to the student to explore the knowledge. And this one, where the teacher explains the concepts. Then these 2 steps, 10th and 11th, give scope to elaborate the concepts which they have learned. And finally, assessments give scope for evaluate the understanding of the children. Thus, this template is aligned with the 5E model also. What about next teaching periods of the chapter? Should we follow the same steps for the second period also? No, certainly not. Certain steps of the flow can be skipped and replaced with some other steps based on the need. For example, all the steps after the greeting and introduction till the model reading, they can be skipped, and in the place of those steps, recalling and connecting the concepts, another new step can be placed. Now let us see the general template for second period of the same concept. If you see here, after greeting and introduction, recalling and connecting the concepts was introduced. So in this step, the teacher asks questions or engages the students in a brief discussion to recall what they learned in the previous lesson. A mind map or concept map can also be used here for visualizing and connecting the previous topic to the new one. This helps the students to see the relevance of the new lesson and creates the curiosity. It activates the prior knowledge for better comprehension, helps the students to make connections between the concepts, provides a smooth transition into the new topic, and encourages the students to participate from the starting itself. What about remedial teaching period or practice period? Commonly, we go for this kind of teaching period after the completion of the chapter for difficult concepts to give more practice. This is almost similar to the second one, but a few more steps can be skipped and more emphasis will be given to hands-on activities that involves the students. Let them explore the concepts, make them understand well, practice well, and reinforce the concepts. In one way or other, we have to engage the children, make them explore the concepts, explain well, elaborate their understanding, and finally evaluate. These 5 E's are very crucial for the success of our class, our students, our school, and our educational system. Now let us consider an example of teaching a challenging concept through a model lesson. Heredity is a significant chapter in class 10 biology, and it includes certain complex ideas that often confuse the students. In this lesson, we will explore how to simplify these concepts for our students and reinforce their understanding through hands-on, play-based, and enjoyable activities during a remedial or practice period. Learning outcomes. Students will be able to understand the monohybrid cross, identify the phenotypic and genotypic ratios of monohybrid cross, deduce the phenotypic and genotypic ratios from the given data. Let us see the class now. Good morning, children. How do you do? With the previous day's class, the heredity chapter was completed, right? Now, in this class, we will try to recall what we discussed regarding it in the previous classes. Let us clarify the doubts if any. Let us try to understand this chapter much better in such a way that we shall never forget it. Okay? Are you ready? Yes. Let us recall once what we have discussed in previous classes. What is the name of the lesson? Heredity. Whatever the topics you remember, just tell me the names. I will draw a mind map for you. Kaushik, tell me the name of the concepts that you remember. Okay, variations. Rani, inheritance. Anji, Mendel experiments on pea plants. Jerina, monohybrid cross. Paul, dihybrid cross. And Yuma, sex determination, expression of traits. So, let me draw a organization chart to show you what you learned in this chapter. Variations and their accumulation, heredity, inheritance, inherited traits, rules of inheritance, Mendel's contribution. In this, we discussed two important concepts, monohybrid cross and dihybrid cross. Then, mechanism of heredity, how the traits get expressed, the expression of traits. Then, sex determination. There are two different types of sex determinations. One, genetical sex determination that depends upon the composition of the chromosomes. Two, non-genetical sex determination where the sex is determined by external factors other than the genetics. So these are the things that we discussed, right? So to understand this chapter much better, let us do a group work now. Now I have a task for you. The name of the task is recall and review. This is a group activity. So I hope you are seated according to your group. If not, please join your group quickly. Okay? Are you ready? Okay. Listen to my instructions carefully. Here are some paper slips, each with a different topic written on it. One member from each group should come up, pick a slip, and then return to your seat. Once you are seated, unfold your paper slip and share the topic with your team. Next, try to recall what you know about that particular topic. On the back of the slip, write down any questions or doubts you have regarding that topic as well as other from this lesson also. Then discuss the topic with your team. Please focus on the task and avoid any distractions. Remember to follow the ground rules. You will have 5 minutes to complete this activity. I will write the topics on the blackboard for you. The topics are accumulation of variation during reproduction. You can refer page number 90 and activity 8.1 for this. Rules of inherited traits, Mendel contribution, monohybrid cross. You can refer page number 92 and first paragraph of page number 94. Then dihybrid cross, refer page number 92 till 8.2.3 and the last two paragraphs of 8.2.3 till 8.2.4. Sex determination, refer page number 96 and 98. I will write your group names against your topic. Which group got the first topic? Okay, second one, third one, right. Now, you and your group can refer the textbook for the topic, discuss again, and prepare a 6 to 10 points note on that. The time given for this is 10 minutes. All should involve in the discussion, follow the ground rules, don't waste the time. Now each group has to present their topic to the class in 2 minutes. JC Bose group, come and present your topic. Good. Any suggestions from other groups? Okay, give them a big round of applause. Sushuta group, come and present your topic. Good. It will be much better if you mention the phenotypic ratio 3:1. Now, If your doubts get clarified, strike off the doubts written by you on the back of the slip. If not clarified, ask me one after another. Any doubt from the group, Charaka? So from our conversation, it seems most of you need support in case of monohybrid class and its ratios. So let me make it clear. For this, let us play a game to understand the monohybrid class. I need two volunteers for the first stage of the game. Who will come? Okay, Ranga and Giri, come. Dear students, observe carefully. Whenever I ask questions, you have to answer me. So what they are doing, you should observe, right? What determines the traits or characters of an organism? Who can answer it? Good. Factors or genes. Let us consider these chess pawns as factors or genes. According to Mendel, how many factors are responsible for a trait? Good. Two. Let us consider these two factors, pawns, control a trait, and these two, it control another trait. Let us consider. Let us consider black pawn as capital T. So it is capital T now, which is responsible for making a pea plant tall. Similarly, consider the white pawns as a small t. So it is small t, which is responsible for making a pea plant dwarf. Now let us consider Ranga as tall plant having only capital T, and Giri as a dwarf plant having only small t. You both please carry these bags containing chess pawns. Ranga, take this bag. It contains 4 black pawns. Giri, take this bag containing 4 white pawns. When I shout a number, each one of you have to take out one pawn from your bag and put it in a box drawn on this table. Okay? So here are your bags. 1, right? 2, 3, 4. Now see these combinations. Each pair had one black and one white pawn, right? That means one capital T and one small t. What will be the height of these plants with the capital T, small t? Yes, they are tall. This is because of law of dominance. A single copy of capital T is enough to make the plant tall, while both copies have to be small t for the plant to be short. Traits like a capital T are called dominant traits, while those that behave like a small t are called. If a pair of factors responsible for a character are of the same type, then they are called homogeneous. Like this. If the pair of factors are of different types, then they are called heterogeneous. Like this. In heterogeneous condition, only one factor exhibits its character out. Such a factor is called dominant factor. In presence of dominant factor, the other factor gets suppressed. It's called law of dominance. Suppressed factor is called a recessive factor. Recessive factor exhibits its characters out only when it is in homogeneous condition. The plants produced by the cross-pollination of these two pure breeds of plants are called as F1 generation plants. In monohybrid cross, these F1 generation plants are self-pollinated to get F2 generation. See, these F1 generation plants have black and white pawns. That means capital T, small t. So the F1 generation plants have capital T black and small t white pawns in equal ratio. So let us take 2 white and 2 black pawns in each bag again and do the same activity. Ranga and Giri, are you ready again? Come, let us do again. Remember, you should not see the pawns when you are taking them out. You should pick them out randomly without seeing their color. Let us start 1, 2, 3, 4. Now observe this combination. If we repeat this activity for some 10 times, let us consider 7 to 8 times similar combination will come. It depends upon the probability, right? Now observe the combinations. One pair with black and black, capital T, capital T. Two pairs with black and white, capital T, small t. And one pair with white and white, small t, small t. What is the height of the plant with capital T, capital T? Tall, right? Okay. Then what is the height of the plant with capital T, small t? We know it is also tall because of the law of dominance. Then what is the height of the plant with small t, dwarf? So there are 3 tall and 1 dwarf plant. 75% tall and 25% dwarf plants. So the ratio is 3:1. The ratio of plants basing on appearance to the outside is called phenotypic ratio. But when we see their composition, one homozygous tall, capital T, two heterozygous tall, capital T, small t, and one dwarf, small t, small t. So the genotypic ratio is 1:2:1. Got it? Why do I always saying to take out one pawn only instead of two? Right, because if character is determined by a pair of factors, out of which one factor comes from the male parent and other factor comes from the female parent. This is possible only because at the time of formation of gametes during meiosis, two factors or genes responsible for a character get separated, segregated, and pass on each into each daughter cell or gamete. Thus, each gamete carry one factor each from each parent. On fertilization, both gametes get fused to form zygote, where a new pair of factors for the new organism will form. Thus, every organism receives its own pair of factors, one each from each parent. This is called law of segregation. Now, let us see how to solve certain problems which are uh, commonly given in exams. See this question here. A heterozygous tall plant was crossed with a homozygous tall plant. What is the genotypic and phenotypic ratios? We know that homozygous tall means they have only capital T, capital T. So let us show it with black. Heterozygous tall plant means that plant had two different types of factors. So capital T and small t. So let us show it with both the colors, black and white. Now what is the genotypic and phenotypic ratios of the offsprings produced from these plants? Let us play the same game again. 1, right? 2, 3, 4. See now, there are two plants with mixed factors, both the factors, capital T and small t. And there are two plants with the same factors, which are capital T, black and black, right? So, if we see the phenotypic ratio, all the plants will be tall only. Why? Because when they are in heterozygous condition, only the dominant factor will exhibit its character to the out. So homozygous tall 2, heterozygous tall 2, but all are tall, right? So what is the phenotypic ratio here? 4:0, right? All are tall. Then what is the genotypic ratio? 2 homozygous tall, 2 heterozygous tall. 2:2 means 1:1, right? Now did you understand how to solve this kind of problems and draw the Punnett squares? Instead of pawns, if you mark them with the alphabets like capital T, small t, this is the Punnett square, right? Let us solve another problem now. A heterozygous tall plant was crossed with a dwarf plant. What is the genotypic and phenotypic ratios. Heterozygous tall. Heterozygous tall means two different types of factors are present, right? So black and white, capital T and small t. Dwarf plant. Dwarf is a recessive trait. So it always homozygous in condition. So all are small t, small t's. Then what is the genotypic and phenotypic ratios? Let us play the same game again. 1, right? 2, 3, 4. Now tell me how many plants are tall and how many plants are dwarf. In these two cases, as dominant factor capital T is present, these two are tall, whereas these two are dwarf. So what is the genotypic ratio and phenotypic ratio? Let us see the phenotypic ratio. These two are tall, these two are dwarf, 2:2, so 1:1. What is the genotypic ratio? This is also heterozygous tall, dwarf. There are two categories only, 2:2, so 1:1. So such a simple thing to solve the problems related to genetics here. Hope you understand now how to draw the Punnett squares, how to draw the phenotypic and the genotypic ratios from the tables given, all this, right? Now let us recall once what we discussed today regarding monohybrid class. For this, instead of recalling by asking questions, let me try another way. For this, let me scan the QR code given on the teacher handbook provided by the CRT, or in the textbook we have some QR codes here now. Let me scan this QR code. It contains the e-content. By scanning it, I will show the summarized video of this concept using the interactive flat panel so that you can revise it. Let me assess once, evaluate once what you have understood. See this question on the board. In a monohybrid cross between two heterozygous pea plants, what is the expected phenotypic ratio of the tall to short plants? Heterozygous, heterozygous means capital T small t. Both are capital T small t only. So it is nothing but simply monohybrid cross F2 generation. So what is the answer for this? 1:1, 3:1, 2:2, 4:0. What is the correct answer? That's good. The right answer is 3:1. See this question now. A monohybrid cross involves a study of how many contrasting traits. How many contrasting traits we consider here? 1, 2, 3, 4. How many? Yes, right, one only. Only the tallness we considered, right? The length of the plant we considered. See the third question. In Mendel's monohybrid cross experiment with the pea plants, which generation showed only dominant traits? Only dominant. Which one? Parental generation, F1 generation, F2 generation, none of the above. What is the correct answer? Yes, correct. In F1 generation, all the plants will be dominant because law of dominance, right? Answer this question. If a pea plant with genotype capital T tall is crossed with a pea plant with the genotype small t, what will be the genotype of F1 offsprings? Capital T, small t, small t. So simply like monohybrid cross parental generation, right? So capital T, small t will come. See, right, correct answer. Note down this work. This is homework for you. Prepare Punnett square for the monohybrid cross at home and show that to me in tomorrow's class. Okay, hope you understand these concepts well. Prepare the Punnett square for the monohybrid class. Dear teachers, so this is one of the strategies that I use to teach my children in my classroom. Hope this video is helpful for you. You please discuss the pros and cons of this video in the cluster complex meeting. Prepare much better strategies to deal with this kind of difficult concepts. Thank you.